Hello, everyone, and welcome to the Real Science Lecture Series. My name is Clay Zimmerman, Director of Science and Technical Services for Biochem. Today, we welcome Dr. Jackie Boerman from Purdue University to examine temporal patterns of muscle protein depletion and accretion across lactation and their implications for nutrient partitioning. Dr. Boerman will also discuss recent findings on muscle dynamics across parities and their implications on metabolic efficiency and production outcomes.
So we're going to get started on talking about muscle depletion and accretion across lactation. And I just want to start with this slide, which I think I show almost in every presentation when I talk about muscle. And I just want to start with talking about the transition period and talking about how muscle mobilization is just a normal adaptation to lactation. So there's a lot of adaptations that cows have when they go from gestation to lactation, and the mobilization of muscle is just one of those. So cows are going to mobilize muscle as a way to prepare for lactation, to mobilize amino acids in order to prepare for lactation, just to meet the amino acid needs and even the energy needs that they're going to require for lactation. So this is a list, and it's not even a complete list of all of the changes that occur when a cow goes from gestation to a lactating state. So when we think about the cow, and one of my students is currently doing a meta-analysis thinking about what the cow is comprised of and the body composition of the cow. But when we think of a cow and what is she made of, the best studies that were done are serial slaughter studies. And from those, we can really get an idea of the body composition of a cow. For Holstein cows, when we think of what they're comprised of, the composition is going to be between 90 and 125 kilograms. And from some of those studies that were done, when we are depriving nitrogen as much as we can, they can mobilize about 25% of their protein. And there's some variation in the amount of protein that cows are going to mobilize. And I'll show you coming up that some of that variation in pattern may be based off of parity as well. Adipose is going to have more variation in how much there can be in cows. So in really under-conditioned cows, they could have less than 100 kilograms to greater than 200 kilograms. That's going to be dependent on body condition score. And those really heavy cows can mobilize in excess of 100 kilograms. When we're visualizing cows, subcutaneous fat is going to be highly correlated to body condition score. So when we're looking at cows and assigning body condition score, we're really looking at subcutaneous fat. And the good news for us is subcutaneous fat is really highly correlated to whole body fat. So even though we can only visualize subcutaneous fat, that's going to be highly correlated to intramuscular fat and omental fat. So even though we're assigning body condition score just with subcutaneous, it's pretty representative of whole body fat. However, muscle depth and body condition score are only moderately correlated. So when we look at body condition score, it's only moderately correlated to how much muscle that they have. We have a graph up here that's from a data set that we collected at Purdue University. So we can see here that this is even a little bit better relationship than what was reported previously, but we have body condition score on our y-axis and muscle depth on our x-axis. And there is a relationship that exists, but it's just not as strong as with body condition score and muscle depth and back fat. Again, cows are going to be in a negative metabolizable protein balance after parturition. That is normal. That is why cows have to mobilize protein. So this is classic work done by Alan Bell. So we have MP balance here. So cows are going to be in a negative MP balance after parturition. They may reach their most negative around 7 days in milk, and then it may take them up to a month before they get into positive MP balance. So during that time, they are going to have to mobilize muscle in order to meet their requirements. So this is, again, normal. The severity of negativity and the duration that they're in negative MP balance is going to be dependent on things like intake, milk production, and milk protein yield. I'm going to show you one of my favorite studies coming up. So these are a set of studies that are done by Larson and others. So because cows are in a negative MP balance in early lactation, they did some abomasal infusions of amino acids that were either casein or amino acids that had a profile that was similar to casein. And again, because the degree that cows are in negative MP balance is going to vary dependent on days in milk. In early lactation, they infused more. So they infused 700 grams really early in lactation, and then 500 grams, and then 200 grams. And they saw no difference in intake. So even though they were infusing additional amino acids, they saw no difference in intake for these cows. However, they saw a really large increase in milk yield when they infused additional amino acids into these cows. And they saw that through 30 days in milk, and they saw a greater milk protein yield as well. So these are the graphs that are going to support that. So again, no difference in intake. However, MP supply increased because they were infusing additional MP. And you see the increase in milk yield. So to simplify this, if we're adding additional amino acids in early lactation, the mammary gland is really selfish. So it's going to take those amino acids, it's going to take them and put them into milk and into milk protein. So I'm interested in muscle. So I started thinking about how would the mobilization of muscle that would contribute to the amino acid supply available to the mammary gland contribute to milk production in early lactation. So that's a thought that we'll get to later on, but I want to leave you with that thought for now. So Alex Tabby and Bill Weiss did another thing because it's not maybe practical to do abomasal infusions on farms. So instead of doing abomasal infusions, they fed additional MP in early lactation. And they did this because they thought that maybe MP and amino acid supply may be of greater importance to early lactation cows rather than energy, because cows may be able to mobilize more adipose tissue rather than protein in early lactation. So they took 40 primiparous cows and 40 multiparous cows and fed them a common diet prepartum. And then they fed 4 different diets for 25 days after calving. And then they had a common carryover diet after those 25 days. So those diets were, first of all, it was a deficient MP diet, which is probably really common to what everyone is feeding out there. So 17% crude protein, you can see the MP levels, about 1,850 grams per day of MP. Then they had an adequate MP that was primarily soy, that was 20% crude protein and 2,300 grams of MP. Then they had an adequate MP diet that was primarily rumen-protected amino acids, 20% crude protein and 2,470 grams of MP. And then a diet that was a blend, but less forage NDF, that was, again, about 20% crude protein, but about 2,480 grams of MP. And you can see that the MP balance was a little bit different in these, but the black was definitely the lowest in MP balance and not very dissimilar to what Alan Bell was showing, that it is lower in early lactation. And as the cows continue to eat more, they're going to reach close to zero by about 4 weeks in lactation. And they actually achieved positive MP with the gray diet, and then they were pretty close, or at least less negative, for the green and the red diets. And I'm not going to go too far into these results, but I do want to focus on some of these. So on the top solid lines are going to be the multiparous cow results. And I want you to just focus on this. There are different responses for the multiparous cows. So the best treatment here was for the green diet, which was the blend, but with less forage NDF. But that's not the same case for the primiparous cows. So we want to focus on that there were different responses in mature versus primiparous cows to MP supplementation in the fresh period. So I, again, focus on that conclusion, and we'll follow up with that moving forward. Going back to the tissue stores that the cow has to be able to mobilize as she's transitioning from gestation to lactation. The first that almost no one talks about is glycogen. That is mobilized really quickly, and there's not a large reserve of glycogen. The second is going to be adipose tissue, and unless the cow is really underconditioned, this is going to generally be the largest pool that the cow has to draw on. And then protein is going to be mostly from skeletal muscle, and it likely starts before calving, with the caveat that it's going to be dependent on a couple of factors. The first paper that I read that really talked about when protein mobilization starts was this Vanderdrift paper. And they indicated that protein mobilization is actually starting in the weeks before calving, and it's ending in about 3 to 4 weeks into lactation. Whereas fat mobilization is starting around calving, but is going to extend much longer into lactation, at least to about 8 weeks or so into lactation. And that is backed up by some of the body composition work, meta-analysis work that one of my students is doing currently, and some of the work that I'm going to show you coming up. So how we are measuring muscle mobilization is using a couple of different methods. The first, and this is maybe a crude way to measure it, but it's a cheap way to measure it, is we're going to use ultrasound images. So we're going to measure ultrasounds across time. So we will measure the longissimus dorsi, which is going to be looking at the loin, and specifically it'll be the eye of the ribeye. And we're going to measure across time the change in the muscle depth. And the reason we do that is we can measure across time, does that change? Does the depth of that muscle change across time? And we can visually see that, but we're going to take an image of that and then freeze that image and then be able to measure that with a program called ImageJ. We can measure it really precisely. And there's a screenshot of the image up here, but we can see from the white line across the top there is the fat, and then we can go all the way down to the edge of the longissimus dorsi muscle. The second measurement there is 3-methylhistidine. So 3-methylhistidine is going to be released when muscle fiber is degraded, and it can't be reused to synthesize protein. So that's going to be released when muscle is broken down. It can't be reused, so it's going to tell us how much muscle is being degraded. And then creatinine is another thing that we can measure in blood, is an indicator of muscle mass. And then finally, we can measure the ratio of 3-methylhistidine to creatinine to indicate how much muscle is breaking down per unit of muscle mass. So the higher that ratio is, the more muscle mobilization is occurring. So taking all of these things together tells us how much muscle is being mobilized. So we're going to look at 4 studies all together to tell you kind of what is happening with muscle mobilization around the transition period. So first, to orient you to this, on our x-axis, we have, again, muscle depth at 1 month prepartum. So this is a month before they're calving, how much muscle they have. And we'll start down here with just the range of how much muscle they have. So we have some cows that have 2 centimeters of muscle, and we have some cows that have over 7 centimeters of muscle. So there's quite a bit of variation in how much muscle cows have about a month before they calve. And then on our y-axis, we have the change in muscle depth from 1 month prepartum to 1 month postpartum. So in that 2-month period that we could define roughly as the transition period, how much change is there in muscle depth? And we can see that everything that is above this 0 line, these cows are actually gaining muscle. So we have cows that have very little muscle depth are actually gaining muscle during this time point. And we have cows that have a lot of muscle depth are able to mobilize more muscle. So if cows have more muscle, they mobilize more muscle during the transition period. And if they have less muscle, they actually have to gain muscle during this time. I'll just point out one more thing on this slide. 3 of these studies are done with multiparous cows. So those are, I apologize if you can't see the colors clearly, but 3 of these studies were done with multiparous cows. The last one is a blue circle, was done with primiparous cows. And you'll notice that the primiparous cows are all grouped towards the right-hand side of your screen, and they're all from about 5 centimeters to 7. So it appears, at least in our population of animals, that those primiparous animals had more muscle depth. They tended to have like on the higher end of muscle depth, whereas our multiparous cows, they had the spectrum of muscle depth. They had a wider range than our primiparous cows did. When we think of all the uses, like why are cows potentially mobilizing muscle? Like what could that muscle be used for? Let's go through it. So constantly, muscle is going to be broken down and built up. It's a constant cycle. So we have skeletal muscle up here. So it's constantly going to be broken down and to amino acids and then built back up. So that is a cycle that is occurring. Once it's in this pool of amino acids, those amino acids could be used for digestive tract requirements. If that animal is pregnant, could be used for fetal requirements, for milk protein synthesis. Those amino acids could be used for TCA cycle intermediates, could be used for, if they're ketogenic amino acids, for ketones, if they're glucogenic amino acids, for glucose, for ammonia and urea. Depending on the amino acids, they could be used for hormone signaling. Specific amino acids we know are immune system modulators. And then we also have metabolizable protein that is going to go into the amino acid pool. So skeletal muscle can be a storage place for these amino acids that they can draw on at different points of their lactation, which I should be able to show you in the next couple of slides. This was one of the first studies that we started to look at about how dynamic muscle is in the transition period. So in several of these studies, we split cows based off of how much muscle they had at study enrollment. So in this case, we enrolled cows 35 days before calving or before expected calving. And then we split them into high muscle versus low muscle. So you see that there's quite a bit of range in how much muscle they had. Our high muscle group averaged about 5 centimeters. Our low muscle group averaged about 3.5 centimeters of muscle. And then they're doing different things here. So our high muscle group is chugging right along. They might be losing a little bit of muscle. And then after calving, they lose more muscle. However, our low muscle group is actually gaining muscle during the dry period. And then after calving, they're losing muscle. So what this looks like, because we are measuring from 35 days before calving to 60 days after calving, is that our high muscle cows lost 1.5 centimeters of muscle, and our low muscle group lost about 0.5 centimeter of muscle. But in reality, our low muscle cows gained muscle during this time point. So they're utilizing amino acids to gain muscle during this time point that they then lose during early lactation. So this muscle is pretty dynamic. This tissue is pretty dynamic. They're gaining muscle at the very end of their dry period for the low muscle group, not specifically for the high muscle group. They're not having a net increase in muscle for the high muscle group. When we looked at some of the metabolites that were associated with this population of cows, we looked at this after parturition. So at 2 days in milk, 14 days in milk, and 21 days in milk, we didn't see any changes in insulin, glucose, BHBA, or NEFA. And we also didn't see any changes in creatinine. Again, this is postpartum, which we should have expected because you saw that those lines came together and we didn't really see a difference in muscle after parturition because those low muscle cows were gaining muscle and became very similar to our high muscle cows. So we didn't see a difference in the amount of muscle that they had. We did see a difference in our 3-methylhistidine, which is our indicator of muscle mobilization. So at specific time points, our high muscle cows had higher 3-methylhistidine and higher 3-methylhistidine to creatinine ratio, indicating more muscle mobilization at specific time points. The other interesting finding here is that our high muscle cows gave birth to calves with greater birth weight. And in this case, it was 7.5 pounds larger. So these calves that were from dams that had greater muscle gave birth to calves that were larger. So 7.5 pounds larger. And in addition, our high muscle cows produce less milk, but not different in energy corrected milk, because a little bit different fat percentage for our high muscle cows, a little bit higher fat percentage for our high muscle cows. So no difference in energy corrected milk. Okay, moving on to another study. So again, we had a little bit higher cow number here, and we're splitting cows again based off of a high muscle and low muscle. And because we're doing this based off of the population we have within that study, our numbers are a little bit different based off of like who's in that study, which cows are in that study. So in this case, our high muscle was greater than 4.6. and our low muscle was less than or equal to 4.6 centimeters at time of enrollment. So we have our high muscle, no statistical difference in dry matter intake or in body weight. There was a difference in body condition score. So our high muscle cows were just a little bit heavier in terms of body condition score. There was no difference in glucose, BHBA, or insulin. So these are all prepartum results. We saw the same thing when it came to the calves, though. Our high muscle cows gave birth to calves that were 8 pounds larger. Then we took some biopsy samples of those calves, and those calves also had greater muscle metabolic activity. So those calves that were born to high muscle dams, when we did a biopsy of their muscle had greater metabolic activity. They also tended to have greater muscle mass as measured by creatinine concentration. No difference in 3-methylhistidine, but they tended to have higher birth weight and greater muscle mass and greater muscle metabolic activity if the calves were born to dams that had greater muscle. We're going to look at some of the muscle measurements from these dams. So again, this is going to be the amount of muscle that they had. And you see that the high muscle cows are going to be losing muscle prior to calving, and they actually lose muscle all the way through from negative 42 days before calving to 28 days in milk, whereas the low muscle cows are going to be gaining muscle during the dry period, and then they lose muscle postpartum. If we break that down into our prepartum results, our high muscle cows lose a net of almost 0.5 centimeter before calving, whereas our prepartum cows gain some muscle before calving. Postpartum, they're losing almost identical amounts of muscle, but total, our high muscle cows are losing more because they're starting to lose muscle before calving. That's allowing some different responses after calving. So we did see that our high muscle cows ate more. So we have an intake response with our high muscle cows eating more. We also saw a fairly large response in energy corrected milk. And then we see that carried on into components. So the high muscle cows produced more fat, protein, and lactose. There was a tendency for increased glucose concentrations for our high muscle cows as well, with no difference in BHBA. And if we look at milk yield throughout the 28 days that we were measuring them, you see that the milk yield response is pretty consistent and follows out to 28 days. We then did another study where we were looking at springing heifers, and heifers are just different. So the other studies that I showed you were all with multiparous cows, and heifers are just different animals. So we again split them into high muscle and low muscle. So we'll start down here with this graph. So we split them into high muscle and low muscle. This is longismus dorsi depth down here. And you'll notice that they started with greater muscle depth. They're not really gaining or losing muscle before calving until right before calving, and then they both lose muscle right before calving. So they're not having a lot of change before calving until right before calving, and then they lose muscle. There is no difference in calf birth weight between these groups. I think it's because they're not mobilizing muscle before calving. And there was no difference in energy corrected milk between these groups either. I think that this population of animals has competing processes. So these heifers are still growing. When animals are growing, their myofibers are increasing in size as they age, but then they're also going through this transition phase where we tend to see this reduction in myofiber size as they are trying to provide amino acids for the process to go through the transition. So these animals are kind of in this phase where they have these competing processes. Heifers also have higher insulin concentrations, which may limit some of the mobilization that is occurring. This is the first study that we measured colostrum weight. There's a lot of variation here, but we didn't see any differences, and we didn't see any differences in bricks either. It's a small population, but this is just what we were able to measure. The student also measured muscle depth at 280 days in milk. We had to measure at 280 days in milk because some of them were very fertile, got bred back right away, which is great, but they had similar muscle depth as they did at 4 weeks of lactation. That's going to come up in a little bit, but heifers don't seem to do exactly the same thing that multiparous cows did. Okay, this is a study that we're probably not going to publish, but I did want to show you. So this is looking at muscle in the gray bars and adipose tissue or fat in the white. So this was looking at first parity animals or primiparous animals versus multiparous animals. And this is if you take your ultrasound and you're looking down into the ultrasound. So we have our adipose tissue and our muscle. And even though in this population we had a 275-pound difference between our parities, we really don't have a difference in the amount of muscle and adipose between these groups, but we do have this decrease in the amount of muscle and adipose tissue as time goes on. So they're both mobilizing tissue, but we don't see parity effects here. We don't see parity by day interactions. So they're both mobilizing tissue. And even though there are differences in body, like differences in body weight, it's not necessarily in the amount of tissue that there is, at least in this population. It's something else that we're not able to necessarily to say that it's differences in the amount of adipose or the amount of muscle, at least in this population. Okay, we're getting through, and we're getting to some of my favorite studies that we've done. So we were interested in looking across the whole lactation to understand what is happening to muscle across the whole lactation. So we split it into segments. So looking at from 0 to 60 days, 60 to 240, and 240 to 60. And we looked at milk production first, and then I'll show you what happened to muscle in the next slide. So in this case, high muscle cows made more milk in early lactation than low muscle cows. And they did that in mid lactation as well, but that switched. And in late lactation, low muscle cows made more milk. For milk fat, just high muscle cows made more milk fat concentration than low muscle cows. And then there was no differences for milk protein other than a time effect. Okay, and then you can see milk yield plotted here. For body weight, this is exactly what we would expect. We have cows that are losing body weight, and they reach their low point around 60 days in milk, and then they're going to increase body weight. But when we get to muscle depth, this is a little bit different. So again, we have our high muscle in gold here, and we have our low muscle in black. Our low muscle are going to our low muscle are going to start lower, obviously, because that's how we're dividing them. They're going to go down, going to be lowest at 60 days in milk, and then They're only going to be significantly different at the 0 time point, a tendency at 60 days in milk, and then significantly different again at 30 days in milk. And it looks a little different, at least to me, than the body weight graph. And then back fat does something I think similar to what is happening for body weight. There's no differences between the two lines. So we see that it reaches the low point at about 90 days in milk, and then it increases over time. So you're increasing and you have a back fat depth that's higher than when they calved. You'll notice that the muscle depth at the end of lactation, you are still lower than when they were at calving. I'm going to show it in another graph to just illustrate the point again. This is when we're combining all of the cows together. So when cows are calving, they have the highest amount of muscle depth. Then when they are going to deplete their muscle reserves until 60 days, and that's when they have the lowest muscle reserves. We're only measuring once a month, so it could be somewhere between 30 and 60 that they reach their lowest point. Then as we do our once a month ultrasounds, they remain largely unchanged, and it's not until the end of their lactation that they start accreting muscle. At 300 days in milk, they still have not reached the point where they have the same amount of muscle as they did at the beginning of lactation. So to me, this says that they are still accreting muscle at the very end of lactation. These cows are on a diet that would support that they have a positive MP balance. They have sufficient amino acids and metabolizable protein to support that they should be able to gain muscle, but it's not in their best interest. So you could say that throughout a majority of their lactation, they are not gaining muscle. They're not net gaining muscle till the very end of their lactation. And when we were doing metabolites for this, I really like this because it showed me something that I didn't think was going to happen. So we looked at this in early lactation at 7 days in milk, at 150 days, and then 300 days. And this is the thing that I didn't think would happen. So we looked at 3-methylhistidine, which is going to be our indicator of muscle mobilization. And it was high in early lactation, which we expected, but it was also really high at 150 days, and it was only low at 300 days. So this tells us that there's really high mobilization even in mid-lactation. So we're still mobilizing a lot of tissue even in mid-lactation. Then we have creatinine, which is our indicator of muscle mass, and our lowest muscle mass is in mid-lactation, which would support what those ultrasounds are showing. And then our ratio of 3-methylhistidine to creatinine is lowest at the end of lactation, which makes sense because they're starting to accrete muscle then. Our NEFAs are high in early lactation, and our insulin is highest at the end of lactation. So this supports what is actually happening, but is not what I expected. So I really like when that happens. So we're going to wrap some of these things up. I'm specifically interested in parity differences, so I'm going to try to incorporate some of those things as well. So from a meta-analysis of 27 studies, looking at MP, like supplementing MP prepartum, they said that additional MP prepartum really benefits heifers. And some of the thought behind that is that maybe that's intake differences or growth requirements, whereas for cows, maybe additional MP benefited cows only for milk protein or only when cows were producing a lot of milk. So maybe there's something to that, but really heifers need that additional MP, and maybe cows are going to benefit more from MP if they're high producing or for milk protein. So there is some parity differences that exist prepartum. And then Tebbe and Weiss also said there's more of a response for multiparous cows with additional MP supplementation postpartum, and the best response was less for GenDF compared with other treatments, and the same response was not for primiparous cows. So this is just to point out that there are parity differences between these groups. You can't feed them or manage them the same. This is some other results that we have seen when we're ultrasounding these cows. These heifers that are turning into primiparous cows, when we're ultrasounding them and when we're observing them, they have more muscle. So when we're ultrasounding them and they are heifers, they have more muscle and they don't lose muscle until right before calving. Our heifers that we are ultrasounding, we're out on pasture. So does exercise have a role in them having more muscle? I don't know, because all of our heifers are out on pasture. And if they transition okay, meaning they don't have metabolic disorders, they eat okay after calving, they lose a smaller percentage of muscle than our multiparous cows. I think that that's because they have higher insulin and glucose concentrations. That's from things that we have measured, but also in the literature. Primiparous cows have higher insulin concentrations. They have higher glucose concentrations than multiparous cows. So that may reduce the need and the ability for those cows to mobilize muscle. Multiparous cows, if they have high muscle, one lactation, the next lactation, they also have high muscle. I say this because we have a relatively small herd at our university dairy, so I tend to enroll some of the same cows over and over. If they're high muscle on one study, they're high muscle on the next study. So it's not that they continue to deplete muscle and they are high muscle as a heifer, and then they'll be low muscle, and then they'll be even lower muscle. If they're high muscle one lactation, they're high muscle the next lactation. But they do end their lactation with less muscle than they calved in. So I think that they're gaining muscle at the very end of their lactation and into the dry period. I didn't show this data, but the reduction in muscle depth is related to a reduction in their cross-sectional area of their myofibers. So muscle is made up of myofibers, and we have measured it through histology, and we think it's primarily type 2A myofibers. That's a detail you probably don't care about, but it is something that we have measured, and we see a reduction in the cross-sectional area of individual myofibers. So it's the ultrasounds, yes, but we also see a reduction in the individual myofibers as well. A little bit more background on muscle in case you are specifically interested in myofibers. So this is from a paper that the first author is Becky Coombe. It was recently accepted by JDS. So myofibers can be stained in order to distinguish between them. Dairy cattle have 3 myofibers. They are type 2X, type 2A, and type 1. So type 1 is going to be our slow twitch endurance fibers. These are going to be primarily our anaerobic respiration. They're going to utilize glucose more efficiently. So I believe that cows are going to preferentially want to have these muscle fibers or more of these muscle fibers in order to have the most efficient transition. So they're going to want to switch between our type 2X into our type 1 or type 2A. I don't know if that's correct, but in order to be a little bit more efficient. And skeletal muscle is responsible for a lot of our insulin-mediated glucose use throughout the body. So it's really important that we have the type of muscle fiber, and we think that muscle fiber switching to more efficient muscle fibers is one of the metabolic adaptations that likely occurs as cows that are transitioning to lactation. So we think that myofiber switching or muscle switching is what is happening. So switching towards type 1 could be what is happening as cows are transitioning to lactation to be more efficient in order to utilize less glucose. Okay, when we're doing some of these biopsies, we're able to differentiate between our high muscle and low muscle cows. And this is just to show you that, yes, we're measuring with ultrasounds. And sometimes that can be like, okay, well, you're measuring it at one time point. But we are able to distinguish this when we're looking at the muscle cells themselves. So I think there's a little bit of, this has some legs on it. So we're able to distinguish between our low muscle and our high muscle. And this was led by Teresa Casey. So we looked at the transcriptional signatures of these muscle fibers. So this is of the low muscle animals. And we saw that for these low muscle animals, we saw that all three pathways that are involved in muscle degradation were actually increased in our low muscle cows. So those are all increased if the cows were considered low muscle. And then we had myofiber types where they had the most of type 2X, then type 2A, and then type 1. So we were able to specifically see the profile of the myofibers. The low muscle cows had increased inflammation pathways, and they also had increased glycogen breakdown. Whereas in our high muscle cows, they had changes in their extracellular matrix. They had a different muscle fiber distribution. So they had more type 2A compared to our low muscle cows. And then they had fiber type modulation, potentially some fiber type switching. And we think that this could matter because maybe they're having more switching as a metabolic adaptation to lactation. So in summary, cows lose muscle. When we're doing those ultrasounds, they lose on average 30 to 35% of their muscle depth. That is not all protein, because some of it is going to be extracellular matrix. Some of it is going to be intramuscular fat. It's going to be like a lot of other things, but they are losing some muscle. On average, they're going to lose 30 to 35% of the depth, okay? But it's going to be dependent on muscle reserves. We have also observed that cows are accreting skeletal muscle at the end of the lactation, and we believe during the dry period, because they end lactation with less muscle than they have at calving. Cows with high muscle reserves, and I'll say multiparous cows with high muscle reserves, give birth to calves with higher birth weights and produce more milk most of the time. Primiparous cows are different, and low muscle cows, they don't mobilize muscle until right before calving, meaning they don't have amino acids, additional amino acids in circulation to allow for amino acids for increased fetal growth and for mammary development. Prepartum, there's some parity effects. So heifers, they're not depleting muscle until right before calving. And primiparous cows also have higher insulin concentrations, which may limit some muscle depletion. So we're going to try to wrap this all up. There's a lot of variation that exists between cows for the amount of muscle they have and the amount of muscle that they can mobilize. So cows that, another thing is cows, we can see on ultrasounds that cows are also depleting muscle. I showed you in early lactation that they're depleting muscle. They're also depleting muscle when they have things like mastitis or lameness. We can see on ultrasounds if a cow is lame or has mastitis, she depletes the amount of muscle she has. Muscle mobilization occurs prior to fat mobilization because they need amino acids at the very end of their gestation. And then in most modeling softwares, we're not accounting for muscle gain during the dry period for transition cows when it comes to MP requirements. And cows with very little muscle reserves are gaining muscle during this time that they draw on in early lactation. And cows with higher muscle reserves use this muscle in late gestation and early lactation for the fetus and the mammary gland. And just to summarize kind of what we're working on next, we're starting to work with commercial farms to have a larger number of cows to look at so we can look at the impacts of muscle on reproduction and health events, and also the role of genetics on the amount of muscle that cows have and the extent of muscle mobilization. So that's what our lab group is focused on in the upcoming months. And these are some of my collaborators. So Dr. Teresa Casey is a lactation biologist who has been working with me from the beginning on this. And then I have a number of graduate students who have worked on muscle projects: Tabby Hurst, Dr. Becca Klopp-Briggs, Dr. Connor McCabe, Kirsten Gouveia, Sam Hanno, Maddie Simons, Becky Coombe, and then postdocs, Dr. Ari Suarez Trujillo, Dr. Linda Beckett, and then numerous undergraduates and the farm staff out at Purdue, and then 2 USDA grants in Zinpro for providing some product for the first study. So I think at this point, I can take questions, and I feel like I talked really fast to get through that. So I apologize if it was fast.
Jackie, that was great. No, great pace, I thought. Excellent job. Thank you. Before we get started with the Q&A portion, we'd like to share a brief video, and then we'll be right back to answer the questions submitted during today's presentation.
Balchem has invested decades perfecting the art of nutrient encapsulation. And now, new AminoShure XL is setting a new standard, delivering 35% more metabolizable lysine than leading competitors. With AminoShure XL, you can provide your cows with a more consistent and higher quality source of metabolizable lysine, leading to improved and more dependable results. A recent meta-analysis found that feeding rumen-protected lysine led to an increase in milk fat percentage and 4.4 more pounds of energy-corrected milk. Feed new AminoShure XL to your cows and have confidence that you're using the most consistent, reliable, and cost-effective source of lysine on the market today. Learn more at balchem.com/XL.
As a reminder, you can still submit questions to the Q&A tab at the top of your screen. So, Jackie, the first question is there a link between muscle depth and colostrum production?
Mm-hmm. Yeah, so we've only measured it in one study. So that was the Primiparis study. And we've also measured colostrum quality. So in the Primiparis cow study, we didn't see anything. We were probably underpowered though. So our thought is that there should be, because if there's additional amino acids that are in circulation from muscle mobilization, our thought is that there would be, and it would potentially affect colostrum yield and also potentially Brix concentration. When we've measured it in studies, we haven't seen anything. So we've measured it in 2 studies now, and we haven't seen anything. So I can't say for sure that there's no relationship there. We're doing it in relatively small studies, and there's a lot of variation when it comes to colostrum yield and colostrum quality. But so far, we haven't been able to observe any effects. And I feel like there should be, but we just haven't been able to observe any effects if they're there. And it would be nice to be able to do that on a larger farm with more cow numbers to see if we can pick up any differences, just because of the amount of variation there is on colostrum yield and quality. So there's a lot of different factors that go into that. So I think you need a larger number than we had in order to pick up any potential differences that could exist.
Okay. Our next question is from June May. And I think I might know the answer to this based off of when you talked about your next steps at the end of the talk, but does muscle mobilization during transition negatively impact reproductive performance?
Yeah, so we're trying to figure that out. That is the next study that we're doing. Our next step is to go on to a commercial farm and to ultrasound 1,000 cows at dry off, at calving, and at 60 days in milk, and then to follow them for their whole lactation, to look at their reproductive records and to look at their health events as well, and also get their genetic information because they would all have been genomically tested. And we hope that that's enough numbers for the reproductive people who may be on the call. But to see if there's any relationship there with the amount of muscle that they're potentially mobilizing. The one thing that I don't know is because the cows that are postpartum, our high muscle and our low muscle cows are mobilizing similar amounts. And a lot of our studies, it's postpartum, they're mobilizing similar amounts. So it would be prepartum. If they're mobilizing prepartum, how is that impacting the follicle development? Like, it would have to be in that phase that we're really seeing differences. And would that impact the follicle development that would impact like that first breeding potentially. So I've had some graduate students trying to come up with a hypothesis on what that impacted. But you could also say, is that low muscle cow that's gaining muscle, would that potentially impact the quality of that follicle during that dry period? I'm not sure. So we're going to look into it and try to have an answer for you, but I don't know for sure right now.
Okay, thank you. So Jackie, specific to, the studies that you've been running there at Purdue, what are the prepartum MP and metabolizable methionine and lysine levels in your prepartum diets?
Oh, shoot. I should have looked that up for you. So MP, and again, it's going to depend on what program you're using. So I'm using AMTS. So, and traditionally, we're just feeding the same diet to everyone. So in our multiparis diets, we're like 1100, 1200 MP for most of our diets is where we're sitting. For our primiparis cows, because our intakes are lower, we're probably below that. So we're maybe shorting MP compared to some of the studies that I have planned based off of the last grant we have funded, is to really bump up MP and to see if we can nutritionally increase the amount of muscle we can gain during the far off dry period specifically. And I don't know if we can do it, but if we can, I think that that's the period of time where the cows are saying they are capable of gaining muscle. I don't think that cows are capable or like programmed to gain muscle during lactation, especially during mid-lactation, because based off of what our metabolites are saying, they're not going to gain muscle. They're going to put those amino acids elsewhere. So I don't think it makes sense to try to have them gain muscle then. I think it makes sense to focus in the end of lactation in the far off dry period, maybe close up as well. So that's where we're going to focus. I don't think in the studies we've done, that hasn't been our focus thus far. So we were, 1100, 1200 MP. And I don't know the methionine and lysine question, but I can look and get back to you.
Okay, thank you. Mm-hmm. Our next question is from Jim Tully. Thank you for a great presentation. Was this presentation similar to the one that they missed at Southwest Nutrition Conference last week?
Yeah, it was very similar. And I apologize. I had the worst flight issues.
All right. The next one is from a presenter we've had before in our series, Dr. Brad Johnson. 2 questions. Great presentation. The first question is a clarification of some data on a slide. The slide where you showed that birth weight was higher in high muscle calves, right below it appeared milk yield was lower in that case, but you said high muscle produces more milk throughout lactation.
Yeah, and I would say most of the time it does. In most of our studies it has. And energy corrected milk in that study was not different, but milk yield was lower in that study. That was the McCabe study. Mm-hmm.
Yeah. And his second...
And can I just clarify that study too, just to full transparency on that study, that was a circadian disruption study too. So we had some other things going on in that study. So we were changing the time that the lights, we adjusted the lights every 3 days by 6 hours. So we were screwing up the people and the cows on that study quite a bit as well. So we were doing some other things in that study. I don't know if that influenced the results at all, but that is different results than we've seen in other studies for multiparis cows. So it hasn't been completely clean all the way through, where high muscle cows have always produced more milk, but there were no differences in energy corrected milk in that study.
Okay. Here's a second question, but I think you addressed it some with your methionine when earlier. So, your comments earlier. Our next question is from Jim Aldrich, and it just moved on my screen. Okay. Obvious question: should we feed more MP metabolized amino acids in late lactation, far off dry period to promote muscle synthesis, and would that do any good?
I think far off dry period, and I think so, but I don't want to tell anyone yes without like doing the research myself. There's some research that we have done with branch chain BFAs, but I for some specific reasons, we have some biopsy data with the role of branch chain amino acids that I still feel like there is a place for branch chain amino acids that I think that, so I still think that there's some role for branch chain amino acids that needs to be explored. And I feel like the far off dry period makes sense only because the demand for amino acids for other places is lowest. So I think the ability for the cows to accrete muscle during that time may be the greatest. So I think that's the time period that I'm focusing on from a research perspective. I think late lactation could also work, but I still think you have the demand for lactation. So I think we may be able to be more efficient if we're doing it in the far off dry period is where I'm focusing on, but we did show that you're able to accrete muscle at the end of lactation as well. I just don't know if you can necessarily, if every farm has a diet that you can specifically allocate there, or if, for that short amount of time. So I think far off may make sense to at least explore. So that's what we're going to try.
Okay, thank you. There are a number of questions similar to this. I'll ask this one from Jeff. Do we know which amino acids are primarily used or needed for muscle accretion and depletion?
Okay. So again, my plug for branch chain amino acids. So muscle is short of branch chain amino acids compared to milk. So I think that if that cow is in early lactation, she could be mobilizing excess muscle in order to meet the branch chain amino acids that are required for milk protein synthesis and milk synthesis. So I think that there could be something there. I don't know for sure. I'm really hesitant because I'm not a muscle biologist, and I think that the more I'm going down this path, the more I'm realizing I don't know, which makes me more excited to keep going and learning more about this. So that's a really good question that, I don't know if I have a good answer to, but I keep going back to branch chain amino acids. I think I'm just kind of stuck on them though. So I don't know if that's the correct answer. It's just kind of what I'm stuck on.
Okay, thank you. Next question from Gayor. What is the relationship between high muscle cows and milk fat percentage?
Oh, so in the study that you saw a milk fat, I think you saw it in 2, that there was a milk fat concentration difference and a yield difference. In one of the studies, there was a body condition score. The high muscle cows had an increase in body condition score. So I think in that case, it was likely that they just had a higher body condition score. So I think it could be some of that they just had a little bit higher body condition score. There's sometimes an intake difference, which could impact yield, but I don't think you're talking about concentration. Yeah, I don't. So I think it could be, there's sometimes higher muscle cows have a little bit higher body condition score because there is, sometimes that relationship there. because it is moderately correlated. So sometimes that exists. So I think with that, you would expect a little bit, at least in early lactation, a little bit of mobilization of those of adipose tissue to increase milk fat concentration. So I think that's likely the case of what's happening there. For yield, I think that there's some intake. The intake definitely doesn't explain in that one study the 14 pounds difference in milk. So on the yield basis, I think the amino acids that were mobilized prepartum could be going to the mammary gland to help in mammary gland development. And then that could help with, if you have increased mammary gland development, you could just have an increased capacity to produce all of the components of milk. It's kind of what I'm going with. But if you're talking about concentration, that's kind of my best guess, is that it's just a little higher body condition score supporting early lactation milk fat.
Do you measure milk fatty acids in these studies?
Not in these studies. We are in some other, not in these studies.
Okay. Does early life or calf nutrition correlate to muscle depth in springing heifers?
I recently got that question, and it's a great question. For this population of calves, no. It could, but like for this population of calves, they're all raised the same. And I got the question, like, could we do a higher plane of nutrition and could that influence it? And that's a great question. I'm telling you, like, the calves that we have, we retain ownership of, and they're on a relatively low plane of nutrition. And they're all fed the same and they're all managed the same all the way through. So we don't have a differentiation as their calves. Like, it's not like they have high amounts of milk and some have low amounts of milk. So I think it's possible that you could influence that really early on in life. And I recently got that question, and it's interesting. Like, could we influence it early on in life by doing like ad libitum feeding to one group and like restricted feeding to another, and then allocate them really early on to differentiate them? In this case, they are managed exactly the same, and that is not what is happening. But it's possible that you could do that early on in life to try to make more. But I think it's likely in this case that it's more genetic than management that is making them be differentiated, because you saw these cows are fed the same diet. Even on the cows that were between 2 centimeters and 7 centimeters of muscle that were on the same study, they were fed the same diet. They were managed the same. And that's the variation that exists.
Okay, thank you. Next question is from Ryan. Have you explored the interaction of back fat loss and muscle loss? Could there be advantages or disadvantages to having a high muscle lean cow versus a high muscle obese cow?
Yeah, we have. So, yeah, we've looked at some of those relationships, and yes. Yeah, so we've looked at some of those relationships and we've tried to like split them out into different groups. I think on any individual study, we don't necessarily have the power to do that, but I think if we combined all of our studies, that would be an approach to do, to then look at the production outcomes. I'm going to write that down. That's a good idea that you just gave me. So yeah, that would be an interesting approach. I think potentially, but I also think like mobilizing adipose tissue is also a normal metabolic adaptation to lactation. So we don't want overly fat cows, but you do need to have some adipose tissue that they are going to mobilize. Like, they need some energy because they are going to be in negative energy balance as well. So there's probably some sweet spot that exists. And I know from ultrasounding those cows that you can't tell visually what a cow is going to be. Like, we have cows that, yes, you can say that's a high body condition score cow, but she has very little muscle. So there may be, yes, because we have the ultrasounds, we could we could split them out and see, but it tends to be that high muscle goes with higher body condition. There is a relationship there. So there's just not like a ton of cows that are high muscle with very little fat, because there is a correlation there. So we may not have a ton of those animals that are high muscle, very little fat. So it may be hard to get a big enough population to follow. But if we combined them all, maybe we could. I'll look and see what our numbers are on, because now we have like 200 and some animals.
Okay. In the interest of time, I'm going to take one final question here, very practical question from Mike Hutchins. How do I determine if my cows are high muscle cows at the farm level?
Yeah, that's a good question. So the ultrasound that we're using is a linear probe, and some farms may have that. So you could just ultrasound, because it's a pretty standard linear probe that you use for repro. So like some farms may have that. And then you could have a cutoff. And so you could measure it like that. Or another thing that I work on that is not really muscle related is video analytics. So we're trying to see if you can, even though we're saying you can't see it, it could be because we're not trained to see it. So we're trying to use cameras to see, is there a way that we can train a camera to see it? So if I feed in the ground truth data of an ultrasound measurement, can I train a camera to see it? The only reason I say we can't see it is because I don't know what I'm looking for. So we know how to body condition score animals because someone told us what we're looking for. I say I can't see muscle because I don't know exactly what to look for. But if I feed in, this cow has 5.5 centimeters of muscle, and then I have 1,000 other cows that I've ultrasounded with that data, I can, with machine learning, train a model to look at images and get better and better and better. So potentially we can develop a camera system that can see and then assign scores. So we're working on that as well. I don't know if it will go anywhere, but that's a potential way that farms can do that in the future. So it'd be similar to body condition score, but it would be muscle depth.
Thank you, Jackie. We have a lot of great questions still out there, but we'll save those for a Real Science Exchange to ask some of those. So thank you, Jackie, and thank you everyone for attending today's webinar. If you have additional questions, please submit them to anh.marketing@balchem.com. The Real Science Lecture Series continues with educational topics each month, including our next few. On February 17th, we'll be back with Walt Cooley on the Real Producer Exchange as we take a deep dive into Skylar Garrick's farming operation in Arizona. And on March 3rd, we welcome Andrew Rose of BioISAC to explore, could your dairy operation survive an attack? Visit www.balchem.com/realscience for details on future webinars and to register for upcoming events. Balchem's podcast series continues to offer a deeper dive into our webinar topics. Log on to your favorite podcast platform and search for Real Science Exchange, or visit balchem.com/podcast. If you want a cool Real Science Exchange t-shirt, just subscribe to the Real Science Exchange and send us a screenshot along with your address to anh.marketing@balchem.com, and we'll get that off to you. On behalf of Balchem and Dr. Boerman, thank you for joining us today.